squeezing the quo. Waddy waddy woo hoo. Almost 300 subscribers. 300 subscribers. Thank you all for their kind, thoughtful, heartfelt comments. Honey bunches, smoochy boochies. Love it. Appreciate you all. Moving forward. Enough of the sap. Let's get back to business. So my last video of the first of the Department of Justice Forensic Genetic Genealogical DNA Analysis, we are going to continue looking at the consumption of forensic samples. We're going to look at forensic samples. We're going to still look at that before we go any further, because we have to fully understand about the forensic samples in order to understand what the hell is going on here. What kind of shit are they trying to pull? So, with that being said, let me show you what I was able to navigate in order to understand for my own personal sanity. So what I found was, after looking at that, I went ahead and I had saw other people were looking at this. And I don't know how anyone can make any sense of what the fuck this is. I mean, I'm sorry, but I started reading it and I was losing my shit. Losing it, because if you had listened to my video last night, even just that small little section about it, this makes zero sense whatsoever. And I was getting so frustrated. My poor cat Milo could feel it and he was being concerned. Yeah, okay. I'm reading this and I'm like thinking to myself, bubblegum in a dish. How many pieces of DNA do you wish? I mean, what is going on here? November 13th, 2022, 4 University of Idaho students were found murdered. As part of law enforcement's investigation to the homicides, the FBI employed the use of investigative genetic genealogy, IgG, using DNA located on a K-bar knife sheath found at the crime scene. Through the IgG process, the FBI constructed a family tree. Why did they jump right to that? If you remember from the video yesterday, you'd get the DNA, and then if you have lacking, which they were going to because of the belt buckle, then you do that. You have to do building blocks. But how many family trees of individuals were on there? Because if you think about it, you have Ethan, Xana, Maddie, and Kaylee, plus the person who did it, who they're saying is Bryan Kohberger. All of their DNA is going to be on that one little belt buckle, and then you had to separate them and figure it out. Well, we know who's at the crime scene, which is the 4 of them. So you can take them out. So how many family trees did you have to do to get to Bryan Kohberger without having his DNA at first? So then the FBI then sent a local law enforcement a tip. The FBI sent local law enforcement as if they are removed from the situation. They were working with them the whole time. And how are they sending them a tip? Are they using the phone number that we all used, that we could use, that they were telling us to call? So they called it in that way? I thought they were working together. If it's from someone on the outside office, like they said that West Virginia, I want to say, was one. He mentioned that there was different FBI locations that were helping. Well, I could understand that for using of resources, but you have people in the police station with them working. You have FBI there. So why are they calling in a tip? I mean, so then he was arrested on December 30th, charged with 4 counts of murder in the first degree. Okay. Nothing about law enforcement use of IgG was used to obtain the arrest warrant for Kohberger or to obtain the search warrant for his DNA. What? What are you talking about? You can only use this for a lead. Okay, so then that IgG lead gave them the Koberger, but then we're not going to use any of that in order for us to get his DNA. You can't have both. You are, just because you're writing it and removing it does not mean that it's okay. Like, this does not make any sense. The state has now filed a motion to prevent the disclosure of the IgG information to the defense. The defense opposes the motion and filed a motion. The defense opposes the motion and filed a motion to compel request and discovery of everything pertaining to the IgG investigation, including the family tree built by the FBI. Okay, so that's the first time I was able to get through all of it, because I had to go through it I don't know how many times before I could like stop going nuts. So after reading that, I was like, oh hell, okay, let's start building our own little case. Let's look into and get our own reference points and resources to see what the hell's going on here. We're going to have to go on a journey. So I went ahead and I started looking, and I found, I'll tell you the order of how I found the things. First it was that. After we had did this, I was like, okay, now I'm going to have to start really looking. So then I found this first. Okay. And this is the National DNA Index System, the NDIS, Operational Procedures Manual, FBI Laboratory, May 1st, 2016. Okay. And this, I just looked at the table of contents here. So it's about quality assurance standards for federal DNA, audit requirements, laboratory, approval of MDS participation, suspension of laboratory from NDS, CODIS administrator, CODIS user, DNA records, federal. Okay, so we'll definitely use this as a resource, as a reference point, as we are learning about this. Linkage of DNA record with criminal history record information not permitted. Retention and removal of DNA records at NDIS. Expungement. Okay, so I'm like, all right, that's a keeper. I'll put my little picture behind it. Let's keep going. So then I went ahead and found this. Okay. And this is the District of Columbia Department of Forensic Sciences DNA report wording guidelines. Okay, so we are getting the FBI's report. And here are the DNA report wording guidelines. Table of contents. Scope, background, safety, materials, okay? Calibration, procedures, sampling, calculations, uncertainty of measurements, limitations. So the scope. Analysts should follow the guidelines listed below when writing DNA reports. Background. To establish the practices for documenting the examination of evidence to conform to the requirements of the Department of Forensic Sciences, Forensic Science Laboratory, FSL, Quality Assurance Manual. The accreditation standards okay, this is huge. The accreditation standards under this and any supplemental standards. When it says accreditation standards, you better fucking follow. That's like JACO, that's all of that shit. So you need to follow that. Okay? Then I end up finding this. And here is September 2016 Executive Office of the President's Council Advisors on Science and Technology. So this is the report to the President of the United States, Forensic Science and Criminal Courts, ensuring scientific validity of feature comparison methods. Okay, so I was like, oh shit, well, here we go with that. Okay. There's who's all involved, and it's all about the PCAST. Okay, so let's go back. The President's Council Advisors on Science and Technology, PCAS, is an advisory group of the nation's leading scientists and engineers appointed by the President to augment the science and technology advice available to him from inside the White House and from cabinet departments and other federal agencies, like the FBI. PCAS is consulted about and often makes a policy recommendations concerning the full range of issues where understandings from the domains of science, technology, and innovation bear potentially on the policy choices before the president. Okay, very good. And that's when I ended up finding this. Okay. And I started reading it. Oh my. It was acting up, and that's why. I think I got like something going on, like a virus or something. Let me know in the comments. Let me know in the comments. All right, so then I end up finding this. And this blew my mind. This is what I started reading last night, and I was going to upload it, and then I had to step back and sleep on it. And it's now 9 o'clock in the morning. I got up and I started looking further. I reread it to make sure, and then I started looking a little bit further, and I ended up finding this guy, which I was like, phew. All right, now I'm back to planet Earth. And then I ended up finding this one, and I felt even better that I was grounded back onto planet Earth. So let's look at this. Let me get back to how it's like that. Okay. This is the National Criminal Justice Reference Service, Office of Justice Programs. It's got all these things going on, which is a little too much for me. It's a little too busy. The authors shown below used federal funding provided by the U.S. Department of Justice to prepare the following resource. Okay? Which I was like, all right. Document title. So right there alerted me. Population Genetic Issues for Forensic DNA Profiles. The author is Carol Rhodes. Now, let me tell you, Carol is pissed. Carol is pissed. And when you're in a scientific field, you can't be, it's a no-no. You have to be grounded. You have to be objective. Like I was saying, as a medical professional, you got to keep your own shit to the side. What's on the inside, your own personal judgments and biases, you have to keep that out the door. When you step in and become a professional in your workplace, you cannot worry. I see people be conflicted with this when they have to take care of people who abuse drugs. You hear them talking, and it's like, you need to stop doing that shit. You need to put that shit to the side. Or if known sex offenders come in, like when I worked in inpatient psych, you need to keep that shit to the side. You have to be objective. You have to treat them as if it's a body. Okay? All that shit is to the side. Yes, you bring in other human aspects to help treat so you can get a complete understanding. But first, you have to look at them objectively. For example, if they come in with a fever, I don't fucking care right now if they abuse drugs, but I have to look at it because they do. Is that a rationale why they have a fever? Do they have an infection someplace? Is it bacterial because of the type of the drugs they use? That's how you look at it. Okay? That's the best practices. That's best practices. So this right here is Carol is pissed, and we'll see why. We'll get into it. It was received April 2022. This resource has not been published by the U.S. Department of Justice. Okay, so the U.S. Department of Justice has nothing to do with this. Okay, This resource is being made publicly available through the Office of Justice Program's National Criminal Justice Reference Services. Opinions or points of view expressed are those of the author and do not necessarily reflect the official position or policies of the U.S. Department of Justice. Boom, right there. So what this is saying, that the U.S. Department of Justice, right there, has nothing to do with the opinions of this author, Carol. Carol's pissed. Okay? Because Carol got questions about her validity. And instead of being scientific about it, she got reactionary. And you know how we know? Well, let's just go through this. So this is the title, Population Genetic Issues for Forensic DNA Profiles. Carol Rhodes, Director, Office of Sponsored Programs at the University of Washington. Okay? At the University of Washington. Where did all these labs, where did they outsource their lab information to do? In Washington. In Washington State. That's where they went. And that's where Bryan Kohberger went to school, Washington State University. Not the university, this is different, University of Washington, but this is the same state, okay? And right here, when I started reading this, I'm thinking to myself, this is not scientific. Accomplishments. Major goals of the project. This project was planned to conduct research on the population genetic issues affecting the interpretation of forensic DNA profiles. The particular topics addressed were interpretation, probabilistic, right there. And of all the other papers we've looked at, you usually have like an abstract, and then you have a hypothesis, and some of them even went further with a null, which I loved. Okay, this is a reactionary piece, and that is not good. That almost becomes null and void to a point. Okay, because it is not scientific. During the award period, we published 36 papers addressing these topics, and they are summarized in the next section. Summaries of publications, interpretation of forensic evidence, response. Okay, interpretation of response. That's not good. Response to Lund and Iyer. Lund and Iyer, 2017, raised an argument. Okay, right there. Raised an argument. That doesn't sound good. Regarding the use of likelihood ratios in court. In our view, their argument is based on a lack of understanding of the paradigm. Don't you question me. Lund and I argue that the decision maker should not accept the expert's likelihood ratio without further consideration. This is agreed by all parties. In normal practice, there is often considerable and proper exploration in the court of the basis of any probabilistic statement. Really? Because we have not seen this up to this point. Ann Taylor had to ask 9 times for discovery. And what was Bill Thompson saying the whole time? Don't know, we're just following the guidelines, privacy. Okay, so there was no exploration going on whatsoever. It was being held tight to chest. We concluded that Lund and I argue against a practice that does not exist and which no one advocates. God. Further, we conclude that the most informative summary and evidential weight is the likelihood ratio. We stated that this is the summary that should be presented to a court in every scientific assessment of evidential weight, with supporting information about how it was constructed and on what it was based. When I initially read this, I was like, holy shit. Carol is pissed. This is not, this is like a fuck you piece. This is what she's doing. And then she goes into the history. So here, as I'm reading, I was like, some of this is okay. I can follow some of this. I have a science base for myself, so like the language. But when it's like wishy-washy a bit, and when you're interpreting things, wishy-washy is not good. That's a sign. That's a flag. That's a red flag. So some of it she does go into that. You know, you got the LRs, which is the likelihood ratios. Some of the things that she was saying, like with the halo. I've heard Vargas talk about Ms. Vargas. So I was getting some of those reference words. Okay. And talking about, right here, the halo type frequencies. And like the lineage markers, the halotype frequencies, okay, the YSTR, okay, and the alleys, so the deconvoluting mixture, okay, so okay, and then the calibrating, okay, and then once you get the two mixed, like when you have a sample, it's going to be mixed, and that's what they said in the court document that we just read, that bubblegum in a dish. How many pieces of DNA do you wish? So you have to separate those samples. And so they talk about that and the inclusive, okay, and exclusive, okay. So it was like understanding bits and pieces, and here the stuttering, the allelic stuttering, okay. And then they started throwing out these numbers, like these percentages. And I'm like, what in the hell are they? Okay. So where are they getting these percentages from? Like, what's the methodology? Like, I didn't see anything on here that said methodology, abstract, nothing like that. Okay. And that is important. In order for it to be considered scientific, you have to have certain sections, and underneath them, that's how you bring about your case of why you are saying that people don't understand it. That's how you have to present it. And we'll get into that further in this next couple pieces that I have. But what I wanted to bring up was these numbers. So here they have 2 times 10 to the 4th power to 8 times 10 to the 6th power. I was like, okay, all right. And then this 2 times 10 to the 28th to 2 times 10 to the 29th. So I had to look that up. And when it comes to nuclear type numbering, 28's not really used. It goes from 27 to, I want to say 29 or 30, but 28's not really in there. And guess what that is? That's the octillion. So this is where they got that octillion from this right here. I'm like, wait a minute. I had to look it up and figure out what is 2 times 10 to the 28th power. I had to break that down. And I'm like, this is where they get the octillion from Carol. From Carol in this, okay? This is Carol's piece about the octillion. And it's a reactionary piece because people are questioning it and she's getting pissed. So I was like, whoa, stop the clock. Okay, now I'm going to have to find other pieces to see if this is even used or if people have talked about this at all. Okay, so that's when I found this, the deconvolution of blood mixtures using Dept separating single cell STR profiling. And where did it go? Right here, right next to each other. And here, YSTR mixture deconvolution by single cell analysis. Okay. But first, let's look at lemon squeezy here. And I was happy that I found this because it brought me back to earth. After reading the Octillion of Carroll's reactionary piece and the court document. And see how it's written already? And this is, it's an article though, but it has a DOI number, .org, which is what you want. Just 2018. Introduction. You see this? Technology. Single cell profile. Study design. Material and methods. Cell separation. DNA extraction. Results and discussion. See how that's done? This is literally a rant. Look at that. Well, I mean, it's just, it's, what is that? It's just a rant. It's not very scientific at all. Let me show you the other one to show. Just to give you this is the YSTR, and this is from Journal of Forensic Sciences. Okay? And yes, it's from the University of National Center for Forensic Science, Orlando, Florida. And I had to take away my biases of Florida. Right now, everyone's acting up. That's Florida. Right now, that's being Florida to the T. So let me just show you this. So you have YSTR mixture deconvolution by single cell analysis. You have your abstract. Okay, you got your keywords. You have your highlights, which is kind of what Carroll did as highlights, but she was just using them as saying interpretation, not highlights. She's just being very, just not being very scientific. Introduction. Methods. Slides. Cell recovery. Direct lysis. You have your, and she didn't even have any of this going on. No type of slide whatsoever. Donor reference samples. She talked about cases and samples, but didn't show any type of statistical evidence. She just wrote freaking octillion on there. PCR product. Stutter ratios. You see how that's done? You see that? Results. Look at, this is what you need to do. If you are upset because people are questioning the validity of what you're doing, then you need to start bringing the fucking black and white and bring the graphs and the statistics. Don't write 2 times 10 to the 28th power as octillion, and that is the reason why. It's because of that. So let me go back to this one first, though. Because this Deconvolution of blood mixtures using Dept array separated single-cell STR profiling. Introduction. Mixture deconvolution. Mixture deconvolution is a main topic in forensic DNA investigations because routine cases involve a lot of mixtures and short tandem repeat STR profiling leads to mixed DNA profiles. Makes sense to me. With the exception of mixed profiles showing a major component or two-person mixtures from whom one contributor is known and the alleles from the second could be deduced. For most of these mixtures, the unambiguous determination of the alleles of a single contributor is not possible. Okay? Therefore, mixed profiles can mostly be used for direct comparison of profiles from known persons. So right there, mixed profiles can mostly be used for direct comparison of profiles from known persons. That is the 4 victims. So what they're talking about doing these family trees, you're doing family trees on unknown victims, unknown people. So where is that coming from? A comparison with national databases, which is the aim of almost all investigations in cases where no suspect could be determined, is only possible for some selected national databases. In general, for searching national databases, single-person profiles are needed or at least preferable. Moreover, mixed profiles are more complicated to interpret and statistically evaluated than a single source samples. In summary, deducing single profiles for all contributors of a given mixture would be a great advantage. For these reasons, several methods were developed by forensic scientists aiming to split mixed stains into their different cellular components. These include the separation of different cell types by laser capture microdissection, LCM, or fluorescent activated cell sorting, as well as the time shift release of the DNA from the different cellular components of a mixture, as the different extraction methods for sperm epithelial cell mixtures, for the splitting of mixed stains consisting exclusively of cells of the same cell type. Some other methods may be helpful. For example, the use of anti-human ABO and CD45 antibody-coating microbeads combined with centrifugal separation for the isolation of white blood cells originating from donors with different blood groups. Okay? As further approach, the sex-specific labeling of cells combined with laser microdissection is used for the isolation of cells from a male and a female contributor. Moreover, some probabilistic models included in statistical software programs were used for mixture deconvolution. So right there, what I just read is how you do forensic scientific paper. Okay, compared to Carol's and comparing to this, do you see the difference? And this is at the Institute of Legal Medicine, Ludwig Maximilians University, Munich, Germany. Okay. So let's go back to here. So this was received July 19th, 2022. Revised, accepted, September of 2022. The Journal of Forensic Sciences. Why STR mixture deconvolution by single cell analysis. Okay. Okay. And the funding is from the state of Florida. Okay. Abstract. Since Y STR typing only amplifies male Y chromosomal DNA, it can simplify the interpretation of some DNA mixtures that contain female DNA. However, if there are multiple male contributors, mixed Y STR DNA profiles will often be obtained. Y STR mixture analysis cases are particularly challenging, though, as currently there are no validated probabilistic genotyping (PG) software solutions commercially available to aid in their interpretation. One approach to fully deconvoluting these challenging mixtures into their individual donors is to conduct single-cell genotyping by isolating individual cells from a mixture prior to conducting DNA typing. In this work, a physical micromanipulation technique involving a tungsten needle and direct PCR with decreased reaction volume and increased cycle number was applied to equimolar 2 and 3-person buccal cell male DNA mixtures, which is what they got from Bryan Kohberger. The buccal, which apparently using the FBI tree as lead, or using those trees, was not used in helping getting Bryan Kohberger or his DNA. Buccal cell male DNA mixtures and a mock touch DNA case scenario involving the consecutive firing of a handgun by 2 males. A consensus DNA profiling approach was then utilized to obtain Y filler plus Y STR halotypes. Okay, so that halotype was what Ms. Vargas was talking about, and Carol did talk about that in her paper, her reactionary piece as well. Buccal cells were used to optimize and test the direct single-cell subsampling approach, and 2-3-person male buccal cell mixtures were fully deconvoluted into their individual donor YSTR halotypes. Single-cell or agglomerated cell clump subsampling from the gun's trigger recovered single-source Y STR profiles from both individuals who fired the gun, the owner and the other unrelated male. Only the non-owner's DNA was found in the cells recovered from the handle. In summary, direct single-cell subsampling, as described, represents a potential simple way to analyze and interpret Y STR mixtures. Okay? So, you got your keywords, and then you have your highlights. And see the highlights? Single cell, stutter, single cell, full single cell. Okay? 2 to 3 person plus. Okay? So, just a little bit further, and then I'm going to end it here. Introduction. Crime scene evidence commonly consists of a mixture of biological material deposited by multiple donors, typically from the victims and or the perpetrators to a crime. When binary mixtures are comprised of both male and female DNA, one approach to obtain probative information about the male donor is to conduct YSTR analysis. YSTR analysis is beneficial as it only amplifies male DNA present on the Y chromosome, and any female contributor's DNA is not amplified. However, if multiple male victims or perpetrators are present, which we only know of one male victim, or perpetrators are present, mixed DNA profiles can still be obtained even with the use of YSTR analysis. In those instances where it is not possible to discern major and minor male contributors, this can cause difficulty in assigning a statistical metric in support of an inclusionary finding that a specific donor's DNA could have contributed to the YSTR mixture at hand. This difficulty primarily arises because YSTRs are inherited from father to son as a chromosomal sized multi-locus halotype block, with all males from a paternal lineage possessing the same YSTR profile. Bearing mutation. and teasing out the genetically linked multi-locus profiles from each of the donors is challenging. Okay? Currently, for example, SWAGDM has not reached a consensus on or prescribes an appropriate statistical approach for YSTR halotype mixtures. Due to the inheritance of YSTRs as a halotype, the use of a Y halotype reference database is required for analysis. In assessing the evidentiary significance of a YSTR profile, an estimate of the rarity of a YSTR halotype in the population is commonly determined by practitioners using the counting method, in which the number of times a YSTR profile appears within the database is used to obtain a conditional probability. Although other methods beyond simple counting YSTR halotypes for single-source male samples such as the discrete Laplace, DL, and Kappa methods have been recommended, which is what Carol was talking about. The Kappa method was in there. While these halotypes databases and the counting method are relatively easily used for the analysis of single-source DNA profiles, their use with YSTR mixtures become much more complex and impractical. Okay? Becomes more complex and impractical. That's why with that octillion, that is not practical. That is not within the realm. That's not within the scope. It's impractical. Especially if distinct major and minor contributors are not present. For YSTR mixtures, several interpretation solutions have been proposed, including, for example, a DL-based approach, a halotype-centered likelihood ratio derivation method, and an ingenious adaptation of the existing mathematical theory of autosomal DNA mixture interpretation to the halotype situation. DNA mixture deconvolution is inherently challenging, even without considering the difficulties described with YSTR analysis, and this is primarily due to overlapping alleles shared among donors in the presence of artifacts, such as stutter, which is what Carol was talking about, that are often tough to differentiate from true alleles. The challenges are further exacerbated with an increase in the number of donors and the presence of stochastic artifacts caused by low template DNA contributors. In order to account for these factors, probabilistic genotyping software is now routinely used in autosomal STR mixture analysis, and it allows for less analysis subjectivity, and considers and accounts for factors such as stutter, drop-in rates, amplification, efficacy or efficiency, and peak heights. However, currently there is no available PG method to address YSTRs, leading to further difficulty in analysis, although some are in the developmental stages. One approach to deconvolute these challenging mixtures into their individual donor constituents is to conduct single-cell analysis by subsampling individual cells from a mixture prior to conducting enhanced DNA typing. The goal of single-cell analysis approach is to retrieve single-source DNA profiles from every individual contributor to a mixture, and has previously shown promise to deconvoluting STR mixtures, as well as some sperm-containing YSTR mixtures. In the present work, we apply direct single-cell subsampling analysis to retrieve individual donor YSTR profiles from equimolar 2-in-person DNA, buccal cell spot mixtures, and simulated touch DNA cases. There you go. So Carol's methods are subjective and it's impractical. So here in that other study, sperm cells were used. This one, sperm cells are used. Do you know why sperm cells were used when it comes to DNA and how that's the best source? Because of the viscosity of it. You know, it's not as fluid as blood. Blood is thin compared to ejaculations. Okay, that substance, it has a lot more viscosity to it. It is thicker. And what's inside of that is sperm cells. And sperm cells are really large compared to a single blood cell. Okay, it's a large cell, so therefore you can get a lot of information off of it because of the size. And it ends up saying and the sperm in itself can be used because it as with blood, it's thin. So with outside environments, with it being exposed to the outside environment, its fragility is there. But when it comes to sperm, okay, it can retain its source a lot longer than blood can on sheets or whatever. So that other paper was talking about sperm. This one was talking about sperm. And therefore, if they're getting their DNA samples, and yes, they use the belt buckle, I'm going to say there's sperm also at that crime scene. There has to be. There has to be. Otherwise, they're just making shit up as they go. So that's all I'm going for right now. I'm going to go ahead and put the documents that I went over. I'm going to put that in the description. And we'll go through the rest of the Department of Justice here about the other aspects here. So that's the forensic samples. Now we'll look into the privacy interests, okay? And then the law enforcement's ability. So let me know in the comments what you think. Until next time, we'll talk to you then.
Touch the ceiling, monks, let's a funk flow. Someone's talking junk, yo, I'll bust them in the eye.